Hello friends, welcome to my YouTube channel, The Microbiology Digest. I am Dr. DP Bora. So today our topic of discussion will be classification and taxonomy of viruses. So in this discussion, we'll be discussing about the DNA viruses. This will be part 1, and RNA viruses will be discussed in our subsequent discussions. Now, what is classification? So classification of virus, it is the process of naming the viruses and placing them to a particular taxonomic system, because each and every viruses, they need to be identified, for which they are required to be placed in a particular system or taxonomy. So that is what actually it is called as classification. So initial periods of virus discovery, we can say up to the first 60 years of virus discovery, there was no system exist, because viruses were totally different from other microbes. So there were no system of classification existed, or the existing classification system would not be applied to the viruses. So that is why, so common systems, for example, common systems were applied to identify or to name a particular virus. These common systems include, for example, the type of disease or the associated disease caused by that virus. So accordingly, the viruses were also named. For example, poliovirus, rabies virus, they were named according to the disease. Then the type of disease caused by a particular virus. If suppose it is a leukemia, then the virus will be accordingly named as leukemia virus. Then site of the body affected. Sometimes the viruses were also named according to the site of the body. For example, those viruses which are affecting but mostly the glands, they were named as adenoviruses. Then sometimes the viruses were also named according to the person or scientist who discovered those viruses. There are examples: Epstein-Barr virus. Then sometimes viruses were also named according to the way how they were transmitted or they were contracted from person to person. So these were the common systems during the earlier periods of virus discovery. These were applied to the viruses for classification or naming. Then early periods, again, there were some classification systems developed. First, comprehensive classification of viruses. It was suggested by three scientists in the year 1962. These three scientists, namely Love, Horney, and Tournier, they suggested the first comprehensive classification system of viruses. Their proposal, or the method they suggested, they included or they adopted the classical methodology or linear hierarchical system, and viruses were classified into phylum, class, order, family, genus, and species. These were the hierarchies of that classification system. So in the present classification also, this part of the system still exists, but entire system could not be adopted. Then comes the ICTV. ICTV, this is the international organization. ICTV stands for International Committee for Taxonomy of Viruses. So this committee is responsible for development of an uniform scheme of classification for virus, and they are also required to maintain the records of all the viruses that has been discovered, and they are to refine or to maintain the universal virus taxonomy, and they will be releasing it to the public domain. So this is the ICTV, International Committee for Taxonomy of Viruses. But this committee was originally formed in the year 1966, and initial name of this committee was International Committee for Nomenclature of Viruses. Then later on, it was changed in the year 1973 to International Committee for Taxonomy of Viruses. So, four principal criteria are used for virus classification, namely the nature of the nucleic acid, the symmetry of the protein shell or capsid, presence or absence of the lipid membrane, then dimensions of the virion. Another classification for viruses was also developed, or it was developed by one scientist, one noted virologist, David Baltimore. This system is called as the Baltimore classification. This was developed in the year 1971. This system of classification of viruses, it is mostly based on the method of viral mRNA synthesis. So according as per this method, Baltimore method, the viruses are placed into 7 different groups, and these groups are written by Roman numerals. It is mostly based on the method of mRNA synthesis and combination of nucleic acid straightness, the sense of the or polarity of the genetic material. All these criteria were used for classifying viruses into 7 different groups, and all these groups are designated by a Roman numeral. So this is the classification of virus by Baltimore method. So as we have already discussed, 7 groups or 7 classes were uh, viruses were classified into 7 different classes. So these classes are written as class 1, class 2, class 3 to class 7. So class 1 includes the double-stranded DNA viruses. So viruses where genetic material is DNA and it is double-stranded. Then class 2, it is the single-stranded DNA viruses. Class 3, double-stranded RNA viruses. Class 4, it includes the positive sense single-stranded RNA viruses. Viruses where genome is a single-stranded RNA and it is positive sense or it is of positive polarity. Then class 5 includes the single-stranded RNA viruses with negative sense genome. Then class 6, it includes the single-stranded RNA viruses with reverse transcriptase or reverse transcription methodology. So this reverse transcriptase enzyme is carried by some viruses, and these viruses are included under class 6. Then the last class 7 includes the DNA viruses or double-stranded DNA viruses where reverse transcriptase enzyme is present or carried by the virus. These are the class 7 classes according to the Baltimore method of virus classification. Next, the present day classification, which is published by the ICTV. We have already discussed what is ICTV. It stands for International Committee for Taxonomy of Viruses. So ICTV has defined the virus species as because the species is the basis for any biological classification system. So for virus also it is same, and the virus species as defined by ICTV as a polythetic class of virus that constitutes a replicating lineage and occupying a specific or particular ecological niche. Okay, so this is a definition of a virus species as per ICTV. Now, ICTV, if we visit the ICTV, this is the website of ICTV, and when we enter to the ICTV homepage, this is the homepage of ICTV, and here all the informations related to virus classification, it is included in the ICTV website. Now ICTV classification, as per ICTV classification, highest taxonomic group for virus is the realm, and each taxa in case of virus classification, it is represented by a suffix. Each and every taxa is represented by a specific suffix which is added to name the particular taxonomic classification for a particular virus. Realm, it is the highest taxonomic group and it is suffixed with the word viria. Then subrealm, it is vira. Kingdom, it is viri. Subkingdom, it is virites. Phylum, it is viricota. If it is subphylum, then it is Vericotina. Class, Vericetis. Subclass, Vericetidae. Order, it is suffixed with the word Viralis. Then suborder, Virini. Family, Viridae. Subfamily is suffixed with the word Virini. Genus is suffixed with the word virus. And the species name, the name of the virus is written here completely. So this is the classification system used by ICTV or International Committee for Virus Taxonomy. Now these are the definitions as provided by the ICTV. It is available in the ICTV website. You can go through how realm is represented, how subrealm, kingdom, how these are classified or how they are represented in the viral taxonomy. So everything is mentioned here in the ICTV website. Now, as per latest release in the year 2024, ICTV has released the master species list. It is written in the ICTV website as MSL. It indicates the master species list of viruses. So these many numbers of viruses are classified by ICTV till the release of this version. Now we'll be discussing the classification of animal viruses by ICTV. So as per ICTV, there are total 9 different orders. These orders, they includes, for example, the first order is the Codavirilis. It includes mostly the bacteriophages. Then Mononega virilis, it includes the single-stranded negative sense RNA viruses. Then Nidovirales, Herpes virilis, Thymovirilis, Picorna virilis, Ligament virilis, Bunia virilis, and Otter virilis. These are the 9 orders of viruses as classified by ICTV. Now, these are some diagrammatic illustration of various viruses and their different properties. First is the RNA viruses, the capsid symmetry, the presence or absence of the envelope, the genome architecture, and to which Baltimore class this particular virus is included. And similarly for RNA virus also, these are the for DNA virus also, the same properties are used to classify DNA viruses, and these are some of the diagrammatic illustration of the DNA viruses. Now we will be discussing the DNA viruses because we will be restricting our today's discussion to DNA viruses only. So first we will be discussing the DNA viruses which are enveloped DNA viruses. So first virus under this is the pox virilis, or sorry, pox viridae, not virilis. Pox viruses are not included to any order, so it starts with the family. The family pox viridae will be discussing. So important characteristics of this family are they are brick-shaped. The viruses are brick-shaped and they are complex in structure. The virus or the virion, mature virion, it measures around 250 by 200 by 200 nanometer in size, and there is one exception in case of one genus of pox virus, that is the parapox virus. So here the shape of the parapox viruses, they are ovoid in shape. Other pox viruses, they are brick shaped, but parapox viruses, they are somewhat different. They are ovoid in shape. And the genome in this family of viruses, pox viruses, it is a linear molecule. It is a double-stranded DNA, and the size of the genome is 170 to 250 kilobase pair. The replication of this virus occurs in the cytoplasm, and these sites where in the cytoplasm the replication occurs in some discrete sites. These sites are generally they are termed as viroplasm. Okay, so these viruses, they have a narrow host range. They are said to be host specific, and they can transfer from one host to another host by mostly direct contact. This is the most common method. Through fomites also they can transmit. through aerosols or sometimes mechanically by arthropods. Then the classification of pox viruses, it is pox viridae is the family. It is again divided into two subfamilies, Cordopox viridae and Entomopox viridae. Then Entomopox viridae includes the viruses, pox viruses, which are principally affecting the insects. So we will not be going into detail of this subfamily. So our discussion will be restricting to the Cordopox viridae family. So again, Cordopox viridae subfamily, it is divided into 8 different genus. Each and every genus is having one type species. For example, Orthopox is the first genus, Vaccinia is the type species, and there are other viruses like cowpox, variola, camelpox, buffalopox, monkeypox, acromegalia. These are the viruses which comes under the orthopox genus. Then second is the capripox. Sheeppox is the type species here. Then other viruses, goatpox, sheeppox, and lumpy skin disease virus. Then parapox, orp is the type species, and other viruses includes the pseudocowpox and papular stomatitis virus. Then Suipox, only one virus, that is the swine pox. Avipox includes the pox viruses affecting the avian species. Then Leporipox, Yatapox, Molluscipox, these are the other genera of Cordopox viridae subfamily. Next family of virus is the herpes viridae. So this is also an enveloped double-stranded DNA virus. Okay, herpes viridae. So this herpes viridae family has now been upgraded to an order. So it starts with order herpes viralis, then family herpes viridae, then rest of the classifications like subfamily, then genus, species. This we will be discussing later. So characteristics of this family, these are enveloped viruses, 150 nanometer in diameter, having icosahedral capsid, and the number of capsomere in herpes virus is 162, out of which 150 are pentons and 12 are hexons or hexamers. Typically, herpes viruses, they have a special covering around the virus. So this is called as a tegumen. This is a layer of globular material. It is present in herpes viruses, and this material is called as tegumen. Then genome consists of a single molecule of double-stranded DNA. These viruses, they replicate in the nucleus, and as they are enveloped viruses, they matures or they are released from the cell by the process of budding. They produce intranuclear inclusion bodies, and typically, herpes viruses, they have the property of maintaining lifelong persistency or lifelong latency in the affected host. Now, this is the classification. It is, as I said, now it has been upgraded to an order. Family is herpes viridae. Subfamily, it has four subfamilies: alpha herpes viridae, beta herpes viridae, and gamma herpes viridae. Then last one is the unclassified. Then alpha herpes viridae is again further subdivided into four different genera: simplex, varicella, mardi, and ilto. Beta herpes viridae, again three genera. Gamma herpes viridae, two genera. Then unclassified is not having any genera. Then these are the type species, and numbers of other viruses are also included in this family and the genera. Next family is the Asparviridae. This is also a double-stranded DNA virus which is enveloped. And this virus is the only DNA virus which can be termed as arbovirus. And the genome of this virus, it is a double-stranded DNA genome, and it resembles the linear plasmid of Borrelia. Then replication mostly occurs in the cytoplasm, but nucleus is required for this process. It is having only one virus, that is the this Asparviridae. It is not having any subfamily. Genus is the Astri virus, and the single species under this genus is the African swine fever virus. So now we'll switch to the non-enveloped DNA viruses. Non-enveloped DNA viruses, first is the Adenoviridae. So here adeno means these viruses are mostly affecting the gland. So characteristics of this virus includes they are non-enveloped, hexagonal in outline, icosahedral symmetry, and this capsid is composed of 252 capsomeres. Then there's a penton fiber which is projecting from the pentons, and these penton fibers are having a terminal knob. This is giving a typical structure like this. And in case of avian adenoviruses, the penton fibers are bifurcated. These are some of the exceptional properties which needs to be remembered. Then double-stranded DNA genome, it is having double-stranded DNA as the genome. Then replication mostly occurs in the nucleus, and virus is released by the cell lysis method. It produces intranuclear inclusion bodies. Next, this is the classification of adenovirus. Family is Adenoviridae, no subfamily. It is having four different genera: must adenovirus, AV adenovirus, ETA adenovirus, and CI adenovirus. And there are some other number of important viruses which are included in each of the different genera. Next family is the Papillomaviridae and Polyomaviridae. These two virus families, earlier they were considered as a single family, and the name of that old family was the Papovaviridae. Papova, it stands for papilloma, polyoma, and vacuolating agents. But now both the viruses, papilloma and polyoma viruses, they are separated by creating two independent families as Papillomaviridae and Polyomaviridae. Okay, so variants, the characteristics of these viruses are icosahedral virion 55 nanometer for papillomavirus and 45 for polyomavirus. Then genome is a double-stranded DNA. Then infection, they are able to produce the persistent infection. Replication occurs in the nucleus for both the families. Properties are almost similar, only the size of the virus is different as well as the viral genome is different. Then classification, Papillomaviridae is not having any subfamily, but there are number of genus or genera are available. Other viruses, there are number of other viruses. Then Polyomaviridae also, the genus, only one genus Polyomavirus. Then other viruses are cited here. Next family is the Parvoviridae. Parvoviridae, this is among the DNA viruses. As we know that there are some DNA viruses with a single-stranded DNA genome. So Parvoviridae is one of the virus having DNA as their genome, but it remains as a single-stranded. Means single-stranded DNA virus. The parvovirus is one of the examples. So it is very small virus, icosahedral, and it is non-enveloped. 32 capsomeres are required to make the capsid. Then DNA is a single molecule which is linear and size is 5.2 kilobase per. They are also capable of producing intranuclear inclusion bond. So parvoviridae, it is having two subfamilies, Parvoviridae and Densoviridae. Densoviridae includes mostly the insect viruses, and Parvoviridae, it is classified into five different genera: Parvovirus, Erythrovirus, Dipendovirus, Emdovirus, and Bocavirus. So these are the different genera, five genera of the Parvo, Parvoviridae subfamily under Parvoviridae family. Next family is the Cercoviridae. So Cercoviridae, again another single-stranded DNA virus, that is, but here the genome remains as a circular genome. Okay, and it is the smallest virus till now. It is the smallest vertebrate virus, DNA virus. It measures only 17 to 22 nanometer. Okay, it is single-stranded DNA. As I said, they are non-segmented, but it is circular. Then these Cercoviridae, they are capable of employing ambissense transcription strategy. This is a peculiar character of this virus. Then replication, it occurs in the nucleus, but the cell cycle, it is in the S cycle, S phase of the cell cycle. and these variants they appear as strings of pearls in the infected cells. This is also another typical character of Cercovidae. So classification, this family Cercovidae, it is not having any subfamily, but it is having two genuses, two genera, Cercovirus and Gyrovirus. Circovirus includes the porcine circovirus 1 as a type species. Gyrovirus includes second anemia virus. Then circovirus genera, it includes some other important viruses like cytosine beak and feeder disease virus. Then porcine circovirus 1 and 2. These are some other viruses which are included under this genera. Next family is the Hepadnaviridae. This is the last DNA virus family, and this viruses under this family they are very unique because they are enveloped DNA viruses. But this virus typically they carries a surface antigen. HBs antigen is carried in the surface. Then genome is again it is exceptional one because part of the genome it remains as a single stranded and part of this genome remains as a double stranded. Means this is a partially double stranded and partially single stranded genome virus. Okay, and this virus although it is a DNA genome, it is it carries the reverse transcriptase enzyme, and the replication of this virus it occurs in the nucleus of the hepatocytes. Okay, so this family Hepadnaviridae is not having any subfamily, but two genera are there, Autohepadna and Avihepadna. Autohepadna includes the hepatitis B virus, then Avihepadna includes the hepatitis B as a type species. Now we have completed the classification of DNA viruses, but some of the important points which are to be considered as very essential. So these are said as viewpoints on the DNA viruses. So we have discussed enveloped DNA viruses, pox variety. Four numbers we have discussed: pox variety, herpes variety, Espar variety, and Panna variety. Single-stranded DNA virus, two viruses we have come across. One is parvovirus and another one is circovirus. Then single-stranded DNA virus which is circular genome, only one, it is the circovirus. Then genome which is partially double-stranded and partially single-stranded, it is in Hepadnaviridae. Virus having DNA virus with a tegumen, it is the herpes variety. DNA virus carrying the reverse transcriptase enzyme, it is the Hepadnaviridae. Only DNA virus which can be termed as herbovirus, it is the Espar variety. Capsomer numbers 162 in herpes virus, 252 in adenovirus. Then pox virus replication, the site it is the cytoplasm, and these sites are specifically called as viroplasm. Then sting of pearls appearance, it is in circoviruses. So these are some of the important points I have listed here, and with this we have completed the classification of DNA viruses. And I again request you to subscribe to my channel if you have not subscribed. I think this information will be helpful for you, so please go through. Thank you very much.